Mach net tows, multiple open and closing net tows, along those same lines. And so at a constant depth of, in this case, 430 meters, the oxygen went up and down. You can see that here. That's where the transect was in this plot. And interestingly, the zooplankton populations responded to that very small oxygen change. So this is, again, like between about 8 and 4 micromoles per liter. Very low oxygen levels, right at the metabolic index of 1. The gray is anything below that where they should not be able to tolerate. Above that, they can. Where the metabolic index in red pops above 1, the abundance of the species in that net corresponding to that point were high. As the metabolic index drops below 1, that species disappears and comes back again. So the metabolic index seems to describe the vertical distribution of these species as well. And while I wouldn't go as far as to call 1 its critical metabolic index, because this is the daytime depth, they've migrated down and they're probably in something of a quiescent state. So in their daytime, they might require more oxygen than that. But. All right. So very quickly, I'll just go through these claims that I mentioned earlier. The first is that there's excess oxygen in polar waters. And where that comes from, if you have an animal in the tropics with a metabolic index of 1, as you move toward the poles, its rate of metabolism declines due to temperature, and the amount of oxygen available increases also due to temperature. And most of the effect is due to the decline in metabolism, but your metabolic index increases to the point where you would have a metabolic index of around 20 in the northern and southern poles compared to very low values near the equator. But that assumes the same hypoxia tolerance, the same physiology, when in fact Antarctic animals, and this is data that was measured by Jose Torres and Joe Donnelly years ago here, at a given temperature, their critical oxygen level is actually a little bit higher than you might expect due to temperature. So the colored points on here are similar zooplankton species. The blue are measurements for Euphausia superba and other Antarctic zooplankton. If you extrapolate those up to a somewhat higher temperature, you see that they quickly reach limiting levels. Moreover, the oxygen actually does decline with depth. This is data from the Long-Term Ecological Research Project along the Antarctic Peninsula. So at about 200 meters, the oxygen reaches a minimum that is very close, even at current temperatures, to something that should restrict Antarctic zooplankton. As you warm, if the Antarctic Peninsula warmed to 10 degrees, they would be right at their critical oxygen level at 200 meters, whereas in the surface, they've got a metabolic index of 3 to 4, which is similar to the critical metabolic indices measured for other species. So they may have a little bit of excess oxygen, but not much. Their evolved tolerances are for very cold water with high oxygen, so they don't have the gill capacity and heart capacity that other animals might have. Whether or not CO2 reduces metabolic scope, you see a figure like this a lot. This is metabolic scope as a function of temperature, and CO2 will restrict it. One of the mechanisms that's proposed for that is that CO2 alters how blood binds to oxygen. And so you would expect they're at lower pH, lower blood pH, their blood would carry less oxygen and they would become more oxygen limited. Oops. So for those of you that saw Matt Burke's defense recently, he modeled that effect and compared it to data collected by Redfield. That's Alfred Redfield of the Redfield ratios fame. He measured up to 3,000 pascals. That's 30,000 parts per million. So it's not something we're ever going to see. Within the range that we are going to see, it's impossible to distinguish an effect within the noise there. So this is that range blown up, and there's no effect of increasing CO2 on the critical oxygen level for these squids. So that's also fake news. Lastly, will warming lead to shrinking fish? That hypothesis is based on the idea that the surface area of gills decreases relative to the volume of animal mass consuming oxygen as you get larger. So if your maintenance metabolism, your resting metabolism is here, you can reach a size of W1. But if the temperature increases, you're going to increase maintenance metabolism, which is going to restrict your body size to this W2. That's the theory. It ignores all of the data which has been collected over many decades that shows that gill surface area or gill diffusion capacity exactly matches metabolic rate. Metabolic rate declines with body mass in the same way. And so the two they're not mismatched, and it doesn't have any effect on body size. This hypothesis would also lead you to expect worse hypoxia tolerance in larger animals, which is not what I see looking at all of the available data. So that's also fake news. And this has been reported as fake news in this paper that takes a little dig and says climate change models need to be based on valid physiological mechanisms, which that one is not. So, all right. So some conclusions very quickly. Hypoxia tolerance is species specific, and it's related to the exposure of a given species to hypoxia. Hypoxia tolerance and aerobic scope are mechanistically linked. Species distributions do appear to be bounded by a narrow range of metabolic index, a critical metabolic index. And most species are operating pretty close to their performance limits. They don't have a lot of room. There's not a lot of excess oxygen. Lastly, any change in oxygen partial pressure or temperature will alter species ranges, will decrease their metabolic scope, affecting energetics and growth, and with ultimate consequences for ecosystems. So, thank you, and thanks to my collaborators, Curtis Deutsch and his group, my students, including Alex Burns, who is not here, Karen Wishner and Chris Roman at URI, Emily Schlesinger is a student with Grace Saba at Rutgers University. She's working on the black sea bass data. Stephanie Gordon I mentioned, and this is funded by NOAA and NSF, as well as this college. And wanted to thank the Global Ocean Oxygen Network. It's a United Nations, UNESCO organized group that is, what are we doing? We're reporting on the issue of ocean deoxygenation around the world. So thank you.